Welcome back to part 3 of the 3-part demo series. In part 1, we went through the bulk RNA-seq alignment pipeline, which generates feature count tables from your raw FASTQs. In part 2, we went through the DGE pipeline, which uses the feature count tables to perform differential gene expression analyses. And in this part 3 of the demo series, we're going to be using the DGE tables that are generated to conduct pathway analyses using the pathway analysis pipeline within the Bridge Horizon platform. To launch this pipeline, you're going to simply click Launch Pipeline within the Pathway Analysis Pipeline widget, which will bring you to the sample configuration page. Now, in a similar fashion to the bulk RNA-seq and the DGE pipeline, to configure your samples, you're going to click this orange button over here, which will bring you to the pipeline sample sheet schema. Now, the DGE file, or the differential gene expression analysis that we'll be using for pathway analyses, is contained in the zebrafish folder. So I'm just going to maneuver there. And right over here, DGE results is the file that I'll be using. To select this file, you're going to simply click this or within this box over here, and then drag and drop to the DGE file column. Now, this DGE file was generated using feature count tables from the pectoral and the somoid tissues, or comparative analysis between the pectoral and the somoid tissues. So for comparison, I'm going to simply write pectoral tissue versus somoid. Since these tissues were generated from the zebrafish, I'm just going to give it a label or project ID zebrafish samples, and then I'm going to select zebrafish from the organism drop-down menu. Now, once you're comfortable with your sample sheet, you're going to click Done, which will bring you back to the configuration page. And as you can see over here, I have one sample configured. To select your pipeline tier, you're going to click Next. Now, it's in a similar fashion to the DGE pipeline, we have a single pipeline tier, a speed tier for the pathway analysis, which should work with all of your DGE files. So select this pipeline tier and then click Next. Now in this page, you can confirm the samples that you have selected, pathway tier, and the estimated time of arrival, and then give it a unique run name. So I'm going to call this zebrafish pathways PEC versus somoid. And then you can click Launch Pipeline to initiate pipeline run. Now, as you can see, the pipeline has initiated one. At this point, we're going to wait for the pipeline to finish before we return back to the demo. Okay, everyone. So the pipeline has completed running. At this point, to go to the output directory, you're going to once again maneuver to Submission Outputs and then click Explore. And this will take you to your outputs. Now, the folders are intelligently labeled. So we did comparison analyses between pectoral versus somoid tissue. As you can see, the output directory is labeled with pectoral versus somoid for the zebrafish samples. So once again, the comparison is merged with your project ID to name this folder so that it's easier for reference down the road. To access the pipeline results, you're going to click this folder and then click Pathways Zebrafish Samples. And as you can see over here, before conducting pathways analyses, we take your DGE list and we separate the upregulated from the downregulated gene lists before conducting pathways analysis for both. So for this demo purposes, I'm just going to walk you through the upregulated pathways. And as you can see over here, we conduct pathways analysis using the Reactome database, the KEGG database, and the Go Biological Pathways database. We also conduct GACA pathways analyses only if sufficient gene lists are able to be enriched. In that case, you will get your GACA plots and other plots such as dot plots and bar plots. But in this case, we generated pathways analysis using the Reactome, KEGG, and Go Biological Pathways. The folders annotated as _data contain your data frames. So just to give you a simple example, if you go into the Reactome data, over here you have a CSV file containing all your pathways analyses. This is a CSV file or comma separated. So it looks a little bit like this. If you open this in Microsoft Excel, you will see that all this data is contained in separate columns. One thing that I do want to point out that we do quite nicely is we convert the gene IDs, which are outputted as default when conducting pathways analyses, and we map them to unique gene names. And this makes interpretation of your pathways and which genes are involved in those pathways a lot easier for your downstream analyses. For each pathways analysis CSV file, we also generate a corresponding MD5 sum. And this is so once you download the file, you can ensure file integrity on your end. In a similar fashion to Reactome, the KEGG data folder contains a data frame and the corresponding MD5 sums, and same thing with the Go Biological Pathways. All your plots are contained in this folder called Plots. And once again, we separate the plots for Reactome, KEGG, and Go Biological Pathways into separate folders. So for this demo purposes, I'm just going to quickly go through the KEGG Biological Pathways folder. There's two separate plots over here. And just another thing to mention is that we generate both PDFs, JPEGs, and SVGs for your output. So for the network plot over here, as you can see, you have an SVG output, but you also have a PDF output, and then you also have a JPEG output, which would be over here. Now, one thing that is quite nice to note is that in this network map, instead of gene IDs, you have gene names annotated for each one of your individual networks, which will make interpretation a lot easier down the road when you're analyzing your data. So this has been a demo of the package analysis pipeline within the Bridge Horizon platform. I hope you enjoyed this demo series. If you have any questions, please let me know.